Okay, and good afternoon and welcome to the Simons Electron Microscopy Center's Winter EM course. We're rounding out our Cryo EM Challenges and Frontiers session with our guest lecturer today, Cathy Lawson from the Institute for Quantitative Biomedicine at Rutgers. And the topic of today's reverse classroom would be on the EMDataBank and a little bit more about structure archiving and validation challenges. So Cathy, thank you for joining us today.
Thank you for inviting me again.
Yes. So to introduce you to our students, you had a very different journey into the EM space. You were a crystallographer and in bioinformatics before getting the EM and before the resolution revolution, before X-ray crystallographers now wholeheartedly go into cryo EM. So how was that transition in different structural biology modalities?
Yeah, that's a great question. So yeah, I was what I would call a hardcore crystallographer for the better part of my career until about 2003, 2004, maybe. I was in an ACA meeting and I heard Bridget Carragher give a talk on how it was becoming possible to automate cryo-electron microscopy data collection. And I happened to have a problem that I was struggling with trying to get crystals of a kind of bad complex that just did not want to form. And I talked to her about it, and she said, sure, make an application and come out to San Diego, and we'll try to image your complex. And that's what we did. I think we went out maybe a total of 2 or 3 times. Turned out stain was the thing that worked really best for us because we were still trying to figure out a lot of details about how to make the complex. But it was a RNA polymerase, DNA, and transcription activation protein complex. And we got a 20 angstrom map out of it, and got some very happy collaborators who we were able to validate a model we had made without having that map available, and it was basically correct. So that was a great result. And then during that time, I was also starting, because I was working with Helen Berman and she had taken over the PDB. I had started working with her a little on some bioinformatics related problems. For instance, she was unhappy that virus structures were it was very difficult to look at the set of virus structures available to PDB and actually try to build a virus. It was just it was just very inconsistent how all the viruses were represented. And so she asked me to help her with that. And working with people at PDB, we came up with a standard representation for matrices that allows you to take the structure that is deposited and actually build the whole virus from it. And then it started to be really interesting to look at EMDB because that started in 2002. So it's kind of a natural segue for me to start working with the people at European Bioinformatics Institute that we collaborated with. And we realized, even though things were looking kind of blobby back then, that eventually things could that this field could become really important. And so we decided to put the effort into setting up the infrastructure for people to deposit structures and to make them available, both the maps in EMDB and the models in PDB. So yeah, so my lab to bioinformatics transition happened around the same time as my crystallography to cryo EM transition. So I never actually did that much with a microscope, although, okay, to be fair, Bridget Carragher and I were graduate students together, and we did hang out in a microscopy course for a year. So that was I don't even remember what type of microscope that was, but it was, yeah, it was a TEM of some kind.
Close enough. So as you were mentioning how the database resources have changed, how have you felt that EM has changed over the course of the years that you've been involved with it?
How has EM changed? Oh, yeah, well, quite a bit. I think I mentioned the blobbiness at the start. And then also, I guess, you know, at the beginning, we didn't really have a good standardized data structure for collecting information from EM experiments. And that's been, I mean, you know, that's evolved, and it's much better now than it used to be. And of course, the structures are, you know, the relative resolution has changed a lot. So yeah, so we're seeing things that are coming out where, you know, very, very clearly you can model residues, base pairs. Whereas before it was mostly, if you had the X-ray structure and you could, you know, try to fit it in to the blobby map, then you could make a model that might make sense.
So you have a lot of structures behind you. Do you have a favorite structure that you always like to look at in the database?
Oh, you mean these? Yeah, those are my, I made a little wall. These are mostly my X-ray crystallographic structures, but I have to say my favorite is always going to be trip repressor, because it was my very first, you know. It has this, it's a dimer structure composed with each subunit composed of 6 alpha helices, and they intertwine like this around each other, and they 2 molecules of tryptophan bind the dimer, and when the tryptophan binds, then the dimer recognizes the trip operator, and when there's no tryptophan around, it's not really favorable for it to bind to the DNA. So it's a really cool story, and I still think it's a very cool story, even though people are, you know, it was a bacterial transcription factor and everybody's all excited about human proteins now, but I think it is a textbook example, literally a textbook example. I've had my daughters bring home textbooks, and I said, oh, I worked on that.
I think people are surprised when you first get a textbook. In some textbooks, then there's a section of like, oh, they're actually primary resources. They're all papers. This chapter came from dozens and dozens or even hundreds of papers. And it's
Right.
just one paragraph.
Yeah. Well, I mean, at a certain point, it's, you know, useful to be able to condense the knowledge into something that is, you know, manageable to, I mean, because if you're going to have a chapter about all of biology, or a whole book about all of biology, yeah, it's you know, you have to be able to make a small summary about it. So
as you've observed EM change over the years, are there any challenges or opportunities you feel that we should be tackling?
Challenges or that you're not tackling?
That we're not really now, or are the same challenges years ago, the challenges we should be addressing more today?
Let's see. So, I mean, I guess the one outstanding thing to me, I know we have half map FSC, model FSC, but it's still a little bit different. So I started in X-ray crystallography before there was R-free. So there was a lot of freedom in modeling back then. And then Axel Brunger came up with this idea: leave aside 5% of your data, and then don't touch it, don't let your model see it, and then at the very end, calculate how well your model agrees with this data that you left aside. I'm just not sure that Cryo EM has come up with anything that is quite as equivalent. I mean, the half map FSCs, I guess, pretty good, but I guess I don't know how much people use this, but you can, as long as you have a lot of images, you can refine your model against one half map and then calculate the half map FSC against the other. There's a couple papers on that. So that's I guess that's kind of equivalent. But it's a very different method. So we've just been trying to encourage experts in the field to think carefully about these issues and try to come up with the best kinds of procedures, so that you can, because when you publish a paper, you want to present information to the readers to help convince them that you actually produced a map that's valid based on your images. So I guess that's the one thing. But I mean, in general, I just continue to be floored by the reach of Cryo EM, in terms of membrane proteins, in terms of large macromolecular complexes, and cryo-electron tomography is really doing some very exciting things now. Yeah, it's moving so fast. I have no recommendations on how further to go in the directions of biology. It's just awesome that's being done.
So let's get into today's topic. Could you give us more of a history of the EMDB and now I guess the EM data resource and how it really started out of the other database initiatives?
Sure. So EMDB was started. It was a European initiative, and let me see if I have I know Jose Maria Carasso was involved with it, and okay, I'm trying to think of, there was a guy in the UK who was a big promoter of it, and I'm trying to remember his name. I can't, it's evading me at the moment. Maybe I'll think of it later, or maybe I'll go, if I get an opportunity, I can go look it up. But so, so the EBI agreed to take on the, creating this initial database, and wrote a couple papers about it. Maybe I can even, why don't I?
It can be open book. There's no reason.
Oh yeah. Well, so so if we say released 2002, Yeah, so these were the first 8 maps that were deposited to EMDB. And it's kind of an interesting set. So there's a ribosome. There's this already, the very first structure was a electron tomogram. And actually there's a lot of subtomogram averaging that was done. So all these PDB IDs that are associated with are actually fitted to the subtomogram averages. And this actually describes kind of the, when you start an archive, you want to say, yes, we want to take everything, even if we maybe don't quite understand how it's all going to fit together. So you cannot, there's no map that you can show that is fitted to these models, unfortunately. But because I talked to Ken Taylor about it several times, and we just never managed to figure out how to do it. But yes, it was Stephen Fuller. So here we go. Yes, he's the one who was a great champion of EMDB.
I think that's an interesting point where a lot of people think of EM now, especially in the lens of cryo EM, but a lot of EM was tomography, and that was a good chunk of what electron microscopy was for people. And then later on, electron crystallography, 2D electron crystallography is when we started pushing the resolution. And then what people think of single particle is just relatively recent. The technique's been around for a while, but the application of what we think of cryo EM now is just really in a few years old. And so that always
true.
like when I'm talking to certain people, I know when they came into EM in terms of the conversation, because there's a big resurgence in tomography, but when I started in EM, tomography was over half of what EM was, and it was a different field and it was a different time.
Very true. Yes.
So how did that reflect on how you started to create this data structure?
So Kim Henrik was in charge of EMDB from the start, and then he was also head of the what is currently called PDB Europe. They had a different name back then, but he and Helen Berman and then the group in PDB Japan had decided that they wanted to collaborate on collecting data for PDB, so collecting structures, and they did, and so they basically set up this agreement where they would collaborate on data in, compete on data out. And that was the beginning of the worldwide PDB. So if you're confused about the difference between PDB and worldwide PDB, PDB, Protein Data Bank, is the archive. Worldwide PDB is the organization that oversees the PDB. And then as a sort of subset of all that, Helen Berman and Kim Henrik recruited Wachu to help to advise on improving deposition for EM structures. And like the first thing that happened was to create a dictionary of data terms. So, like for a microscope, what are the parameters that you would use, to collect your data? What was the voltage? All these various kinds of things. And so that dictionary was created, and then it was integrated into both EMDB and PDB. And this permitted us to make the first joint map model deposition system. This happened in 2008, I think. and I was heavily involved in that. And so I was working with EBI. And at that point, we had a joint grant with NIH, with WACHU as the PI, and we worked very closely with EBI all through this project. And a few years ago, everybody was getting all confused for years about EMDB versus EMDataBank. And because we had been calling ourselves EMDataBank.org or EMDataBank project. But EMDB, again, is the data archive, and we were trying to differentiate ourselves as the project. So the same difference, PDB, WWPDB, EMDB, and we decided to call ourselves EM Data Resource, just to make it really clear that it is not EMDB, but our website now basically hosts a search service that allows you to find EMDB maps of interest, whatever would interest you. So I hope that explains what you were asking about.
So how did you decide to form the deposition system, considering EM has many different modalities within there and the data sets are different? And the standards, I think you were involved in a lot of challenges in establishing, or at least beginning the conversation to establish standards. So you can talk about that, and I guess we'll segue into the latter topic of all the challenges of creating standards.
Yes. So creating standards is it's a community activity. It's kind of like, I've heard it, and people at PDB have sometimes called it herding cats, because you kind of have to encourage people to get together and have discussions. Ideal thing is to get them all in a room for 2 days and lock the door. Hopefully standards will come out at the end that everybody agrees on. So we went through that process for the dictionary, and then in 2010 we were very fortunate to get Richard Henderson's attention and Michael Rossman's attention, and other such luminaries. We're very concerned about making sure that when people publish their papers, they would be able to provide some statistics, some kind of means to tell reviewers and readers, how much can you trust this map, right? How much can you trust the model that you created from the map? And so that was this paper. So I think, it ended up not being published till 2012, I guess. Is that right? Yes, so this paper here. And so that was a real learning experience for me. I think this was really the first time that I had more or less managed the whole process of running that meeting and getting people to agree to write the paper and what we would say in it. So some of the key things were, I think the idea of the gold standard FSC was created by Steve Ludke chiming up during the discussions at that meeting. And then, you know, with some agreement, you know, of other people. So yeah, it's a kind of different, it's a different skill set than running an X-ray crystallography lab or, you know, advising research scientists. It's been a learning experience for me. I'm kind of getting into it. I've also now been doing these challenges. I guess maybe you were going to talk about them at a certain point, but we have, so we're currently running a model challenge. We finished one 2 years ago. It seems to be about, I don't know, there are certain people who think it should be run more frequently, but at least my pace is every 2 years. I don't know if I can manage to do it more frequently than that, but we're running a challenge at the moment where we have a group of people who have advised us, and we've set 3 different maps, targets, and there are models available for each of them. And although we want people to look at the map model fit and remodel everything, there's going to be a particular focus this time around on validating the ligand positions. And so that's why you see here the you know, the different components. So I think that answered your question.
We have a question in chat, Juliana. Would you like to pose your question?
Yes. Hi, my name is Juliana. One of my questions is, what are the mandatory items to be deposited right now?
Yes. So mandatory items. It would be nice if there was just a list of them somewhere. So, because if I take you to the MMSIF website, it's kind of tough to sift through it. But, you know, for instance, you know, for microscope, the microscope model is required. The resolution of your map is required, and how you derive that resolution is required. But that's actually only for single particle. You had asked me this question about different modalities. So there's actually slightly different deposition interfaces for the, you know, for single particle versus crystallography versus electron tomography and subtomogram averaging. And then, I mean, if you're depositing coordinates, there are specific mandatory items for that as well. Let's see, it might be useful. I don't think this quite answers your, not sure how, to what extent this answers your question, but I'm going to share something else in a sec. Okay. Okay. So Bob Glazer asked me to contribute a chapter to a book he's writing. You may have been involved in this too, Ed. I'm just going to show you that book. So this is a book that's specifically for single particle cryo EM of biological molecules. And the very last chapter is about archiving. And sorry, I'm kind of going through this fast. Yeah, so this is what I wanted to point out. I've been thinking that I should actually take this image and just put it up on the website. So we kind of came up with this as a checklist that would be useful for people to prepare for deposition. You know, the most important thing is we really want to know is your resolution because it lets us rank your structures in some way, the structures in some ways. So to prepare for a single particle deposition, you want to have a CCP4 MRC map, your coordinates, preferably in PDBX MMSIF format. If you watched my lecture, I think I covered that last year. The difference, if somebody has a question, you can ask about that. The EMDB image is just some kind of a, pretty, I actually made a point of making a montage of some of my more recent favorites of images. So these are just images that are contributed by the author and they represent your map. And it's what you see when you go to the EM data resource site on our carousel. So make it pretty. And then people will be attracted to it. Optionally, you can deposit half maps. Optionally, you can deposit FSE curves. It's a good idea to, well, you should validate your data beforehand. You should know what the resolution of your map is. And if you want to, for your coordinates, you can use the WWPDB has a standalone server that will allow you to evaluate the quality of your coordinates with respect to bonds, angles, clashes. Of course, you're going to have perfect Ramachandran plots because everybody does. Did I rant about that last year? I might have ranted about that last year. Because it used to be that Ramachandran plots were gave you some independent validation of the quality of a protein structure, but nowadays people actually use Ramachandran restraints, and that totally screws it up. So everybody has perfect Ramachandrans. Statistics. Here's an important thing that you might not think about ahead of time. And it's particularly important if you're depositing coordinates. Every protein chain has to be associated with a UniProt ID. If you have something, unless of course you're depositing something that's totally new and there's no UniProt ID, then of course you can't. But if your protein does have a sequence that is known by UniProt, then you're going to want to give the UniProt ID. Another important thing is to check any ligands. So there's a rather complicated system with PDB. For new ligands, they have to be assigned a new ID. but if your ligand is an existing ligand, then you should assign it to an existing ID. I'm a little less familiar with the details of that, so I'm not sure I could. But there's pretty good advice on the WWPDB site if you run into issues with this. And then, of course, the deposition is using OneDep. So it may have been, I'm not sure that really answered the what are the mandatory items, but at least that answers what are the things that you need to have prepared for deposition. And, hopefully you do need to know basic information about how your experiment was carried out. You need to know, for instance, how many images were used to generate your map. That's a required parameter. There are a lot of non-mandatory items that we love when people actually sit and take the time to make a complete deposition, because that makes the archive structure that much more valuable to somebody who's interested in looking at it.
Thank you. What about raw data or raw images? Are they required?
I'm sorry, what images?
Raw data?
Oh, the raw data. So PDB and EMDB do not require raw data to be deposited. There's a separate archive called Empire, EMPIAR, which will take raw data. It is not mandatory to deposit. I think a lot of people do go ahead and deposit their raw data there. I think the last time I checked, there was maybe about 5% of all maps in EMDB had raw data in Empire. Actually, we can check that also. Going back to here. So I designed this search system, and I basically made sure I can search for things that I'm interested in. So here's an example. So there's 794 structures that have Empire raw data associated with them. So if you're interested, that's how you can find them. Oh, and then actually, if you were looking for something else, like least. So wow, there's already been more than 1,000 maps released this year. Wow. That's amazing. But so, and 15 of them have Empire raw data. So that's interesting. So respiratory complex, and wow, there's some really interesting stuff. I feel like it's candy when I get to look at these lists. It's just every structure is just gorgeous. So alpha-synuclein, there's a whole bunch here. Nice. Okay.
I have another question. When you do the deposition, it generates like a validation report.
Right.
Is that generated with Phoenix or with other program?
That's a great question. So WWPDB has its own software package for creating the validation report. That said, it uses a number of different software packages to do it. Phoenix is, I think, not currently being used, but I think it's being considered to be used to replace what's being currently being used. But Mulprobity, for instance, is used for coordinates. Now, in terms of, what are we using? So let me just take a look here. So if we were to look at the map validation report for this, so this might be enlightening. But yeah, so you can see here, like this is a mandatory item, single particle resolution determination method, microscope model, image detector. And yeah, so this depositor was pretty good with providing some non-mandatory items. So there's a software that was developed at EBI for creating these images, these different images that appear here. So that's not Phoenix. And yeah, again, so this is an EBI software package that does this kind of thing, the map value distribution and the volume estimate and a rotationally average power spectrum and Fourier shell correlation. So, so this is actually a newer version. I'm glad they did this. So they now have the author provided FSC curve, a calculated FSC curve based on, I guess, based on half maps that were provided. No idea why they're so different. Might have something to do with masking. Who knows? If the reported resolution certainly fits the deposited FSC. Yeah, so I guess to answer your question, that Phoenix is not used. It's a software that I don't even think, I think they don't, for some reason they don't even mention. I'm pretty sure they don't mention in the user guide. Yeah, it's because it's homegrown software. But this is so this one is basically the same, but it should also have, oh no, that's the one we were just looking at. This one also has the coordinates. So it has it's pretty good. So it's, like I told you, perfect Ramachandran statistics. Very good. Seems to, I would say this is a pretty good model based on what I see here. So a lot of green. The gray means that it's not modeled. It's part of the chain that's not modeled. Red means that there's the proportion that is, I guess, represented with poorer density. But it's not that much for such a complex structure. And then sadly, when you have a lot of, yeah, this is going to be a really, really long report because every single ligand has a validation on it. But let's see, just go down to the end. So we have this atom inclusion, which was kind of an old idea for how just an easy way to do how well does the model fit into the map. I expect that this is going to go away fairly soon to be replaced by map model FSC. Possibly also EM Ringer, possibly also Q-score, because the issue is the recommended contour level. That is a required value you have to give, and it's there's no, I guess, sort of quantitative way to arrive at that value. It's basically what is the value of the of the contour level that you used when you were trying to interpret the map that was most useful to you, that encompasses most of the model. And that's different for different people. It's different at different resolutions. And so it's but so this is, take it with a grain of salt. At least most of the model fits in the map at the recommended contour level. So I hope that answered your question about software. But we did find actually that, I guess, the Phoenix program has a lot of different correlation coefficients that it uses for deciding or looking at the fit of the model to the map. And in a paper we just recently published in Nature Methods, we showed that those correlation methods and the methods that are used by RefMac and what other programs? MyATOPS, Tempe, almost all the measures that are used for map model fit it doesn't really matter which one you use, just stick with one and use it throughout your process of making your best model. And that statistic will help you decide, did I make an improvement or did I, do something to screw up my model? Whereas if you actually wanted to use these values to compare depositions across structures that were deposited, there's only a few of those are actually useful. So Q-score seems like it may be one, EM Ringer maybe another, atom inclusion definitely not. So it's sort of an interim measure that had been come up with that we will move away from. But most people, for most structures, the model's pretty much inside the map. But you wouldn't believe what I saw when I was looking at maps and models in about 2005. I saw crazy stuff like maps that weren't at the right magnification relative to the model. The map would be tiny or it would be huge relative to the map, to the model. So, that was one of the first standardization issues that we had to deal with when we were working to create a joint map model deposition system. So, so this is an improvement that almost everybody has their model and their map. You would think that that's nothing, but are there any other questions?
So the reported resolution is the one that you deposit or the one that is calculated by the validation report?
Okay, so the reported resolution that gets used, for instance, on a, say on this page here, that's the number you type in to your deposition. It's whatever you type in.
Okay, thank you. And one last question. How long does it take for the deposition to be processed?
That's a good question. I mean, usually not too long. So we have, or I should say the worldwide PDB processes with agreement with EMDB, they process the EMDB entry and the PDB entry together if it's a joint map model. And there are 3 sites. There's soon to be a 4th site. PDB China is coming soon. And your deposition will be handled by the regional group that handles, depositions from your country. So the US handles all of the Americas, North America and South America. PDB Europe handles European entries, and PDBJ has been handling all Asian entries, but I think they're going to split it with China. Each group has a team of annotators. I think RCSB has the most. I think these days we're like at 8, something like 8 annotators. And so it just basically depends on their load. They keep track of something called a magic number, which is the number of depositions that have yet to be processed, and they try to keep it down. But I think usually, they'll get to your deposition, within a week, I would say, typically, of you, for initial processing. And once that's done, you'll get notified to go and look at the entry following annotation, because they look to see if it needs corrections in terms of the particular. Apparently the ligand is the most problem. If you have ligands, it's going to take longer. If you don't have ligands, then the annotator will be very happy and will be able to process it probably in about, a very short amount of time, like, an hour or so. Yeah. Yeah, so I would say, a week turnaround or so.
Thank you.
Mhm.
Cathy, do you have some sense of how long, if you have all the materials for deposition should take?
Yeah, in a way, it very similarly to the annotation side. In some ways, it depends on how complicated your structure is. So, okay. These poor people had to basically enter all of these, 54 different. They basically had to specify all this chemistry for this complex. It's, in ribosomes. But I guess there are ways that you can make it easier on yourself. So MMSIF format, there's a tool called PDB extract that you can use that will move your structure from PDB format to MMSIF format And you can also enter all the sequence information there, and it will produce a MMSIF file that you can deposit. I guess right now, what's the rule is, so if you have a crystal structure, you are required now to deposit MMSIF format. They haven't made that true yet for EM or NMR, but it's a matter of time. But yeah, I would say this business, the whole business about defining the different components of a structure takes the most amount of time. And then if you contrast that with, let's see, I happen to know about this. This was the structure I was referring to before. It's my one entry in EMDB. Here, we were not at the point. So this was deposited in an era where we were just basically allowed to define freely define subcomponents of your complex, and they didn't necessarily have to match up with all the polymers that were defined in PDB. So yeah, so RNA polymerase hollow enzyme. If you went and looked at the PDB structure, there's 6 different polymer entities that have to be described for that. But here it's just very simple. But nowadays, you would have to define all if we were to deposit this now, we would have to define all the polymer entities, both for the map and the model. So.
So you're very well adept at navigating the website and using the searches. Are there any tips and tricks on how to use the search? Because you wrote most of the search here.
Glad you asked that. So I actually have, I wrote a little news item in 2020, at the end of 2020, what's new? Because I actually did make some enhancements to search, and I thought it'd be useful to let people know about that. So one thing is, I was sitting around at home trying to figure out how can I contribute to the coronavirus, blah, blah. So I created a specific search for coronavirus structures. And so that gets automatically updated every week, and it's just so it's basically a what would you say? It's a curated view of my regular list, which is for everything, because I've added all these little buttons here that you can, so you could just look for structures that are spike. Structures that contain ACE2 receptor, structures that contain ribosomes. There are actually such things. Those that were made by subtomogram averaging, those that have empire data, those that have coordinates, and those that don't. So I'm actually pretty proud of this. That it does seem to be, other people have told me they found it useful. So I reckon if you had to find it, so if you're home, you can just go COB and enhanced search. Here's another thing that is new, because sometimes it's hard to really know, what are the most popular data values. And so what I did was for, I guess, some of the top kinds of data items that people might want to know about, like microscope model. So now I have a list. So I mean, you can show all the terms if you like. So there's a total of 65 types of microscope models. We have an enumerated list in the deposition form. So you have to pick an existing value. And if you have a new microscope, you can always ask for it to be added. But then it actually goes through a process where somebody asks me, hey, should we use this name, or would you suggest a different name? So and so each of these, so if you're interested in, which structures were solved using this microscope, you can click on this link and it will send it to you. What other things? So the primary citation journal, the most popular is Nature Communications. Interesting, Nature, PNAS. Reconstruction method. Obviously single particle. The sample species, most popular value is null. I can't say. I don't know why. Second most popular is Homo sapiens, though, but that's pretty cool. Specimen type. Again, this is a enumerated list, so you have to pick one of the values. Vitrification instrument. Voltage. This one's kind of interesting. So did you know that there's actually structures that have been done at 1,000 volts and 2,000 volts? So here's the three that were done at 1,000 volts.
Yeah, Japan. I'm surprised there's a megavolt up in Albany. I'm surprised that didn't make the list.
Oh, well, maybe eventually. And different kinds of energy filters. Here's a case where we need to clean this up, obviously, because we have GIF, quantum, GIF, bio, GIF, quantum. This is a future. Cathy, please sit down with this list and come up with a more sensible enumeration, please. That'll be one of my jobs the next year, probably. Imaging detector. This list is maintained pretty well. Yeah, see, there's 1,000 structures that were solved using film that were deposited, Kodak SO4, or 13 with a different kind of Kodak film. So you can get that information. Reconstruction software is available. Symmetry imposed. Most popular value is C1. This I created because of the specific interest that we were having about I was trying to find maps that had interesting ligands. So simply for that purpose, I set up the capability to find ligands. So if you want to know which structures have waters modeled with the coordinates, then you can click there. Which 333 have ATP ligands. And this was an interesting one. So somebody approached me and said, I would like to know all of the entries in EMDB that have half maps. I said, oh, okay. I don't have the capability to do that, but that's pretty easy to implement. So this was like within the past, I think about a month and a half ago, I added this. And so not just half maps. So there's 2,000 or 2,800. But if you're interested in knowing structures that have layer lines, there's 3 of them. Not very recent, but they're there. And yeah, there's 97 that have structure factors. I think for the most part, these are structure factors that, well, it looks like there's a lot of electron crystallography. All right, so yeah, so this is my, like one of my new, this is my power tool. And it's you know, you go to a shopping website like Amazon or something, and you'll see off on the left-hand side, it has all these little widgets where you can you know, refine what you want to search for. And I don't quite have that. This is much simpler, but I still think it's pretty effective. And I guess the other thing I would want people to know about is that this kind of list is pretty to the eye, which is why I made it this way, because it has these nice, and it's very compact. But the actual original search that I created can be accessed here. And this search allows you to download the results. So this list, you can't download the results, but you can just interact with the results. But this one, you can add different columns. If you're interested to know, let's say, a DOI of the primary citation, maybe what method, what are the fitted model ligands, and update the results. And you get a list, and you can look at all the rows if you like, or if you're ultimately interested in working more with these particular entries, you probably just want, you can choose either CSV download or Excel download. And copy works even too. If you hit copy and then you open a spreadsheet, you can just paste. So we've been pretty happy with the way this search works. Gotten some good feedback about it. If anybody can think of something that they would like to be able to search in EMDB, we could check it out. And if I can't do it, then it'll give you something to work on. Any suggestions?
So I guess to start wrapping up, what are the challenges for you, or how do you see the EM data resource will be expanding over the next few years, especially with the data explosion and I guess more tomography is going to come back.
Oh yeah, I bet. Absolutely. So, so we will likely be continuing to run challenges at least every 2 years, maybe more frequently. Just to, it's an opportunity for the community to come together and have discussions and think about what are the important things we need to think about in terms of validating structures. And in terms of, there's one other thing I can show you. I just finished there's another article that will be coming out soon in the Journal of Biological Chemistry. And just want to show you one of the figures from that, because I think that'll be, might be kind of it, relates to your question. All right, stop share. And I'm just going to share this one. So this is one of the figures. It's actually going to be a collection of review articles in the Journal of Biological Chemistry on the occasion of PDB becoming 50 years old. In case you don't know, there is a symposium that will happen in May 4th and 5th, a 2-day symposium. You might be interested to sign up for it if you go to the WWPDB page or any of the PDB pages. You will find a PDB 50 information that you can sign up. It's free. It's free to participate. All right. So this shows you for EMDB and PDB EM entries, where we've been. So there are actually a number of structures in the PDB that were from EM, either electron crystallography or electron microscopy, starting with bacteriorhodopsin from Richard Henderson way back when. There are actually up to 20 structures by 2000, which you can't see on this scale. But then here we go, a pretty rapid explosion with maps and EMDB and models in PDB. And just for fun, I decided to extrapolate the curves. And here I have total number of entries predicted, and this is a log scale now by year. And so again, here's PDB, EM, coordinates in PDB from EM, maps in EMDB from EM. So eventually they will become equal. But PDB X-ray, I also extrapolated. And so let's see, in another, what is this? So in another 15, 14 years, Cryo EM is going to overtake X-ray crystallography, is my prediction, based on this. And could happen sooner. Who knows? And NMR has will probably continue on. It has its niche, but it's a different kind of niche than what Cryo EM can offer. And Cryo EM offers a lot. So, great. So that's, I guess, what I have to say in response to that question.
And if there are no other questions, we thank you, Cathy, for joining us. We can stop recording.